Hi everybody, welcome to the second lecture in the Pharmacogenomics Elective, PHA 5110. Today we're going to do a refresher on the previous concepts that you've learned on pharmacogenetics in previous courses so that we have a nice solid foundation for the remainder of the course. We'll talk a little bit and I'll introduce to you the concepts of pharmacoepigenetics, and then finally we'll go over some pharmacogenetic databases that you'll be using throughout the rest of the course to work on different assignments and clinical case scenarios. So first off, to review, what is the definition of pharmacogenetics? And pharmacogenetics is essentially the study of the effect of genetic variation on drug response. And that drug response, of course, can happen in two different ways. The most common way we think of is drug response, as in the beneficial responses, so this would be the good side. And then the other side we think about is side effects. So this would be the bad side. And genetic variation can influence essentially the way drugs work on our body and influence the rate at which we have a good response and a negative response. And then a lot of times in pharmacogenomics, you'll hear it interchangeably used with pharmacogenetics. So what is the difference? So genetics really refers to individual level, smaller, either a one gene or few genes, while pharmacogenomics, and you think of it this way, genomics is much more broad. So this is population level, a term for population level study of pharmacogenomics on drug response. And really it's much broader. And a lot of times, it's not with one or a few specific genes. It's looking at genome-wide changes and their effects on drug response. So those are the actual two terms. Again, this is the much smaller term. You can think of it like this. And this is the much broader term. However, in the end, they're both used interchangeably. And I usually say pharmacogenomics, but sometimes I might say pharmacogenetics. So in the end, there is a difference, but usually people use one or the other or switch between the two. Now let's go over a few essential terms that we're going to talk about essentially every class. First of all, what's an allele? And an allele is an alternate form of a gene at a specific location. So what kind of alternate forms are we talking about? Well, we're really talking about mutations, variations, and all those types of terms that we talk about even in the first lecture, like homozygous, heterozygous, dominant, recessive. These are all terms that are referring to alternate forms of a gene at a specific location within that gene. So how about diploid? What does this mean again? And that means you have two copies of each gene because you have two copies of each chromosome, one from mom and one from dad. And then finally, kind of similar to allele, what is a polymorphism? And a polymorphism actually has a very specific definition, and it's an allele or a variation that occurs at greater than or equal to 1% in the human population. And there's many different types of polymorphisms. Some are called SNPs or SNPs. I usually call them SNPs. And these are single nucleotide polymorphisms. A few other ones are VNTRs, and these are variable number tandem repeats. We'll see an example of that in a little bit. You can have gene deletions. You can have CNVs, which are copy number variations. Essentially, it's in the name. You have a different number of certain regions of a gene. And one thing that's common to all polymorphisms, especially when they are SNPs, is they're searchable by what's called the RS or reference SNP number on the internet. And I will be showing you several databases where you can input directly the RS number to find information on a specific polymorphism. And one thing to keep in mind, even though the definition has to be in greater than 1% in the human population, there are a lot of polymorphisms throughout the genome, and there are many, many, many. Okay? We're talking millions of SNPs throughout the human genome. Now, do they all make a difference on certain disease states and certain medication response? No, not necessarily, but it's good to know that human variation or human genetic variation is very common, but whether or not it's consequential, well, that's the point of pharmacogenomics. So let's start to go through some examples of SNPs or SNPs, those types of polymorphisms or variations where there's changes within the DNA. So let's go through the first one here, and let's do the example where here we have a C, okay? And that would code for the amino acid glycine. And as we first learned about in the first lecture, again, everything's coded based on a codon chart. And you worked a little bit on this in your homework as well. And essentially it's that codon chart that really tells the machinery in the cell how to read the DNA. And really it's reading the RNA, right? Because the tRNAs work with the RNA to grab amino acids and then create a polypeptide, which eventually creates that protein. So let's say in this synonymous SNP, we have, instead of a C, which is here, we have a T here, which goes to here. And so what is the effect? What happens in a synonymous SNP is nothing. So the definition of a synonymous SNP is that you have a variant that leads to no change in the amino acid that's being coded. And what is this really benefiting from? It's benefiting from the redundancy of the codon chart. Without that redundancy, then we probably have a lot more errors that would be detected. Which is a non-synonymous SNP. So let's give an example of a non-synonymous SNP here. So that would be something like we have a TAT here, which codes for a tyrosine amino acid in this polypeptide chain. However, we end up having a non-synonymous SNP where the A is changed out for a G. So in its place, there's a G. And what does that do? This one actually does make a change where it changes that cysteine, changes that tyrosine, I should say, into a cysteine. So it actually causes a functional change. And when I say functional, I'll say this word a lot when we're talking about pharmacogenomics. Functional just means it actually causes a change in the amino acid, which can potentially cause a change in how the polypeptide forms, on how it folds, and then ultimately the function of that protein. Okay, so far we've talked about two SNPs or SNPs. So that variation where the DNA alters by one letter. The first, where a change in that letter didn't cause a change in the amino acid being coded for. The second, the non-synonymous, where it does cause a change in the amino acid that's being coded in, which could potentially cause changes in the protein. What are some other potential causes of SNPs? So let's take this one, for example. Say we have a G here, and that codes for tryptophan. And the SNP here is a G, right there, to an A. And what does that A cause? That actually ends up causing a stop codon. And so this is what we would call a premature stop codon SNP or a truncated protein SNP, something along the lines of essentially saying that this SNP causes a massive, very bad change in how the coding of the DNA actually makes into the protein. And this is an issue because obviously if you have a truncated protein, the likelihood that it has any proper function is almost zero. And we do see these even with CYP enzymes that we'll talk about later. So let's take a break real quick and talk about why some SNPs may be important and some may be less important. And that comes down to the way that DNA is processed into RNA. So if we recall from last class, what we had was strands of DNA. Okay, and DNA was double-stranded. And the dark lines here refer to exons, while the dotted lines are introns. And so, when it gets changed into RNA, through the various processes that we talked about, at first the RNA is faithfully copied to include both the exons and the introns, right? But then RNA goes through its own pre-processing step that includes that spliceosome that we saw. And what happens is that what comes out are the introns, and what stays in and what's spliced together are the exons. So you get one solid, nice looking piece here of RNA, and then we have, remember, the cap that's added on here, 5-methylguanine cap, and the poly A tail. And then that is transported out of the nucleus and goes and interacts to make, eventually, a protein. So let's go back to where SNPs occur. So if you had to hypothesize, where would the more important SNPs occur and potentially the less important SNPs occur? So if you had a SNP that occurred here, in this exon, obviously that's going to be carried through and it's going to end up here on your RNA, which then, depending on what type of SNP it is, synonymous, non-synonymous, stop protein SNP, it could have a pretty substantial effect on the protein. However, if you had a SNP here, for say, in the intron, that one's going to actually end up being taken out here by the spliceosome. So theoretically, when you have a SNP and an intron, the likelihood that it actually has a functionally important effect, remember functional being changing that protein somehow down the line, is very, very low compared to having one that's in the exon. You at least have some sort of chance that it might cause some sort of new amino acid to be put in there, or a stop codon, or something that causes a change in the protein's function. And later, just to throw a wrench at you, we'll talk about some exceptions to that. So let's continue on in talking about some other versions of polymorphisms that can occur in the human genome. So the one we'll talk about in this slide is called tandem repeats or copy number variations. And again, as I mentioned before, it's really in the name. So it's a change in the number of times something is repeated within a particular gene. So the example that we're going to talk about here is in the UGT1A1 gene. So there's two different types here we'll talk about. We'll talk about UGT1A1 star 1 and UGT1A1 star 28. So they both have similar endings, or at least the major portion of their genes, where they have an exonic portion here, and they actually have the same number of exons. And here, what I'm drawing is each of the boxes represents an exon. So let's number the exons. So in this particular copy number variation, this isn't what actually differs between them. What differs is the actual variation in the promoter region. And as we learned about in the first lecture, the promoter region is critical to where the actual transcription starts, so where the RNA polymerases recognize the gene and begin to initiate transcription. So let's draw out these differences. Okay, and so where these differences are, so on this UGT1A1 star 1, which is considered the wild type allele, the difference is that the T and the A in the promoter region repeat only 6 times. So that's captured here in the promoter region before we start getting into the exons and the introns of the gene. The difference is that with the star 28 allele, so the alternate version of this gene, we have an extra copy of the TA in that series of TA repeats, so a copy number variation. And that's depicted this way with a TA parentheses 7, meaning that it's TA times 7 before we get to the body of the gene. So what does this end up causing? Well, when you have a wild type, right, you just have normal function and expression. But with the alternate one, with just the extra T and A in it, they've found that you end up having decreased expression and altered function. So just that one extra T and an A in the promoter region decreases the expression of this gene, alters its function so that UGT1A1 is not able to do its job very well. And we potentially will see an example of this in pharmacogenomics later in the course. Okay, so the last one we'll talk about, the last polymorphism, is a gene deletion. And the example we're going to talk about today is CYP2D6 star 5. So that's a specific allele version of the CYP2D6 gene. And in this one, what they have found is that it's not just a SNP, but it's thousands of bases within the gene that are actually deleted out. And what occurs when this happens? This is considered a loss of function polymorphism, meaning what's in the words that the CYP2D6 enzyme just doesn't work at all. Okay, and we'll see the consequences of that many times in several cases as we go through this course. And phenotypically, what does this look like? You're considered a poor metabolizer, which we'll learn about in a few slides. And it affects many different drugs because about 20% of drugs or more are metabolized by CYP2D6. So what does this actually look like if we were to draw out the gene itself? So let's call this star 1. And this is the star 5. We're going to use the same symbols we used in the last slide, where the boxes are the exons. And what happens is that this exon on the right is actually completely removed. And so what you have is a truncated transcription of RNA, which leads to a non-functional protein. So why is this all important? Because functional genetic variation leads to alternate phenotypes. So here's an example of an altered phenotype based on the metabolism of CYP2C19. So in this figure, we are looking at omeprazole, which is metabolized by CYP2C19 into 5-hydroxyomeprazole, which is actually considered the inactive form. So that's kind of the simplified pathway of omeprazole being inactivated in the body. So what can we see in this graph of this study by Burghardt and colleagues? What we see here is that poor metabolizers, which would be this area, have much, much greater area under the curve, which area under the curve is more of a scientific measurement of plasma levels as compared to the squares of the circles, which are both heterozygous and homozygous respectively. So in other words, if you are a normal metabolizer, so the circles, you would have what would be considered the population average drug levels for omeprazole. If you are heterozygous, meaning that you have intermediate function of CYP2C19, you have about intermediate levels of the omeprazole active metabolite in your body as compared to normal metabolizers and poor metabolizers. And then, obviously, as we go up the scale, you have completely loss of function or a very low functioning CYP2C19. You're going to have very, very high levels of omeprazole because your body just cannot metabolize that and then remove it from the body. So this shows us that genetic variation has very potentially drastic effects on drug response, and namely phenotypes, which is another name for trait. So previously I mentioned that there's going to be a caveat that I was going to talk about, and here is that caveat. So, not all synonymous SNPs are necessarily silent. And I'm going to show you an example of that. So we have a SNP in the ABCB1 gene, which is a transporter gene, and the SNP is called 3435C to T, and we're going to learn in just a few slides what this actual nomenclature means. And what happens is that it essentially does not cause any amino acid change because it's synonymous, right? So let's look at what it looks like in terms of the letters. We have an ATC here, and that codes for an isoleucine. And then in the synonymous SNP, we have an ATT here, which also codes for isoleucine. So no change in between them. It's again that silent synonymous SNP. And really we would expect there to be no effect on the protein function and then therefore the phenotype of this genetic variation. However, what they found in studies is that ABCB1, this specific SNP, is associated with decreased mRNA levels and a decreased risk of efavirenz-associated hepatotoxicity. So very interesting. Not only is there a molecular change from a synonymous SNP, but an actual clinical decreased risk of a side effect from a drug. And so what do they think that the actual cause is here? They hypothesize that it's something to do with mRNA stability. Now, how this is happening, we're still not sure, but I'm going to give you a few ideas in the next coming slides on how a synonymous SNP can potentially be not so silent. So what are some possible causes of a synonymous or an intronic variant not being silent and actually having a functional consequence? And that's because of this funky term called linkage disequilibrium. And linkage disequilibrium really refers to how the genes or the chromosomes mix amongst themselves during meiosis, during that step of recombination. So let's first talk about a concept called linkage equilibrium. And for this, we're going to draw out two chromosomes, a red and a blue chromosome that work together in their dyad, and on them they have 4 alleles in total, 2 alleles per chromosome, an A at one end, a B at the other end, and then on the red one it has a little a and a little b. So what happens in linkage equilibrium is that the chance of these chromosomes mixing during recombination is entirely equal because the event occurs in an independent random fashion. So let's draw that out here as well. So you can see the chances that you'll end up with big A, big B in the recombination event, little a, little b, big B, or big A, little b, is 25% for each because, again, it's occurring in an independent random process. Now, let's compare that to what we're talking about here, which is linkage disequilibrium. And again, we'll draw out the same chromosomes and we'll see where the differences occur. So again, we have a dyad here, except for the difference in these two scenarios is that the alleles are much closer physically on the chromosome. We have a big A and a big B up towards the top of chromosome, blue one here. And then we have little a, little b at the top of chromosome, the red chromosome here. So this process occurs in a non-random fashion. Therefore, you don't have an equal chance of inheriting either gene. So let's see what that looks like real quick, and then we'll explain why. So what happens is that you have the chance of inheriting, right, just the big A, big B. You have a, little b. And then here, because they're so close, you might have a big A, little b, and finally a little a, big B. However, in the recombination event, because they're not far away, like in the linkage equilibrium scenario, we don't have a 25% to inherit each one, because the recombination actually is occurring physically too close. So it ends up that these two pieces are mixed at a non-random rate because they're so close. So it would be very highly unlikely that the cutting of the DNA occurs right between the alleles. Compared to the linkage equilibrium scenario, you have a lot of room in here to cut the chromosomes and mix them, and you don't have that room here. So that's what linkage disequilibrium means. And another way people put it is essentially that you have some sort of SNP throughout the genome that's actually highly correlated with another nearby SNP in the genome. So when we think about it in terms of the synonymous or the intronic SNP not being silent, what's happening is an indirect association. So let's say we have our DNA here, and we have a SNP here, but this SNP is actually synonymous. It's actually in linkage disequilibrium, that's the term they use, in linkage disequilibrium with another SNP that is actually non-synonymous. So what's happening is the indirect association is really a tagging of the synonymous SNP onto the nearby non-synonymous SNP. All of these variations that we've been talking about so far can actually be inherited together to make up not a genotype, which would be a single SNP or variation, but a haplotype, which is actually a pattern of sequential SNPs found on a chromosome or a gene. And so technically and generally, these are very tightly linked, aka in linkage disequilibrium. So recombination's very rare. So it's kind of hard to understand and really imagine what a haplotype is. So what I'm going to do is set up a scenario here of a haplotype. So let me write that out for you. So imagine that we have a single gene that we're looking at called gene ABC. And this gene, we're going to look at it in 4 different people. So here's our 4 people. So let's write out what this gene looks like. Okay, so what I've set up here for gene ABC in 4 different people is their sequences. So you can see the non-varying nucleotides are in black throughout the sequence. The ... dots mean that the gene extends this way, and then there's more non-varying nucleotides in here. There's more non-varying nucleotides between here, and then the gene extends this way as well. And so I've captured 3 different SNPs here, red SNPs that can be either a C or a T. This second blue SNP is a G or an A. And the third green SNP is a G or an A. And that's for 4 different people. So let's set up their haplotype now. Okay, so now what I've put in here are 4 haplotypes based on these 4 people. Haplotype 1, haplotype 2, haplotype 3, and haplotype 4. So what these haplotypes actually are, right, so you can see here's the 3 SNPs that we originally started with. However, they're capturing additional SNPs that go this way. So this would be the purple going this way. These are all the SNPs that way within gene ABC. And then all the SNPs on the right side are captured over here in gene ABC. So you can see there's a pattern here of SNPs that give 4 distinct haplotypes. Okay? Then this can actually be utilized to do what's called SNP tagging. So in this case, we would take this one here, this one here, and this one here. And we would genotype here for either an A or a G, a T or a C, or a G or a C, in order to determine the 4 haplotypes that are available without having to do genotyping of the entire gene. Because again, if this is what the 3 SNPs look like, just the area I wrote out here, imagine that this goes thousands of base pairs to the left in order to capture all these SNPs, and thousands of base pairs to the right to capture all these SNPs. So that can get really expensive really quickly in genotyping cost. So instead, if you do tag snipping, you can look at just these 3 areas, because if you have an A, you're either haplotypes 1, 2, or 4. If you're G, that'll pin you to haplotype 3. For this one, you can do a T or a C. If you're T, you're either haplotype 1 or 3. If you're C, you're 2 or 4. And then this one, if you can do a genotyping of G or C in this position. And if you're a G, that means you're a haplotype 2. So it helps us segregate. And likely you'd have to add on another tagging SNP in order to look at haplotypes 1 or 2. But again, if you're doing the snipping here, if you had an A here in this haplotype and a T here, it would be essentially that you're a haplotype 1, right? Because it's A and T. This A has a C here, so that means you're not that haplotype. Comparatively, if you're looking and you're only looking at C's here, you could be a C or 1, 2, or 4, and so then you'd have to genotype this one too and look at your combinations of SNPs. But in the end, this pattern of sequential SNPs, so all these SNPs put together, are made into haplotypes, which we will see later in the SIP genotypes. A lot of them are actually composed of haplotypes, where they look at a series of commonly inherited SNPs that are really rare in terms of recombination, so they're almost always inherited together in order to determine SNP genotypes. So all this genetic variation that we're talking about can actually be inherited in a certain pattern in order to have a certain distribution across the population in general. And somebody actually created a mathematical model for this and called it Hardy-Weinberg equilibrium after the people who created it. And Hardy-Weinberg equilibrium has some certain assumptions, such as the population in which you're doing the prediction of distribution of gene variation has to be quite large. It has to have low mutation rates, so your genetic variation has to be really what the genetic variation is. There has to be no new introduced genetic variation through mutation. It has to be randomized mating, okay? And there has to be no real selective pressures being put on the inheritance of that gene. So let's do an example here. So we have genotype AA, AB, and BB. And you can also think of it as in terms of the alleles, with having a wild type, or the dominant genotype here, the heterozygous wild type, mutant type, and the recessive mutant type. So what's the actual equation here that we use in order to determine Hardy-Weinberg equilibrium to see if a population is in Hardy-Weinberg equilibrium, but also to predict the distribution of the genes across the population. It's a pretty straightforward equation, and it is p squared plus 2pq plus q squared. So let's do an example here. We have a population. We have a population whose allele frequencies for A and B are 70% for A and 30% for B, respectively. Go ahead and calculate what the distributions of the genotypes would be in this population. So it would be 0.7 squared plus 2 times 0.7 times 0.3, because we're doing frequencies, right? So you would divide it by 100, plus 0.3 squared. So in order to get our answers, we go ahead and do the math for each one separately, and then multiply it by 100 again to get us back to percentages. And so the numbers of, right, A, B, B would be 49%, 9%, and then 42%. And there's two other definitions. There is the percent of people that have the A allele would be the, they don't really have a term for it because we generally do the next one I'll tell you, but it would be what would be the major, quote unquote, frequency, the major allele frequency. What we do have an actual definition for is the percent of people that have the little b allele, which would be the minor allele in this case. We actually call that the minor allele frequency, which is shorthanded MAF. So you might see this, you'll definitely see it in the databases that you'll look in and possibly in papers as well. So the population distribution of gene variation can be modeled as p squared plus 2pq plus q squared. And from that, we're able to tell and calculate a minor allele frequency for any given gene, as long as we have population numbers to start with. So why are all of this genetic variation, why is it so important? And that's basically because your genotype combines with environment and patient-specific factors to give you your phenotype or your clinical trait. Some people call it observable trait. For pharmacogenetics, generally we call it drug effect. And so I always put in here, it's always genotype plus environment plus patient-specific factors, because there are instances where maybe one of these is entirely or almost entirely a predictor of phenotype. But by and large, it's always a mix. It's always a multifactorial input in order to get phenotype. So especially in pharmacogenetics, although we're looking at genotypes, we never forget about the patient's environment, and we never forget about patient-specific factors. Talk about the intersection of pharmacogenetics with drug mechanisms. So let's go back to our central dogma, which started with genes, went through mRNA to get proteins, and now let's add in the drug portion here. Proteins have effects on pharmacokinetics and pharmacodynamics of drugs. So when you give a drug, it has to go through its pharmacokinetic effects. So that's the effects of the body on the drug. And then through its pharmacodynamic effects, which is the effects of the drug on the body, to get some sort of response, whether it be positive in terms of the efficacy that we're looking for in the drug, or negative in terms of side effects. So now, where do pharmacogenetics come into play? And that's up here with genes where they can have genetic variation. For example, you can have variation that causes increases or decreases in the actual gene product Or you can have variation that causes dysfunctional or hypofunctional product. And again, these go back into making proteins themselves. However, they can have actual effects on PK, can have actual effects on PD, and so can an increased amount of product can have an effect as well on PK and PD. And it's better to think of this, if you want to get rid of the mRNA portion, really think about this as your protein product or your hyperfunctional or dysfunctional protein that goes and intersects with the drug PK and PD. So you can see the intersection of pharmacogenetics with PK and PD really occurs through the changes in the genes that cause changes in the proteins, which will inherently affect how the body treats the drug or how the drug treats the body. So let's zoom into the intersection of genetics with drug treatment, specifically looking at metabolism, which would be PK. So most all drugs, I would say, go through some sort of metabolism, almost all drugs. So some of them go through metabolism in order to be removed from the body, to be excreted, and some are actually activated, right? And we call those prodrugs. And the main types of metabolism are phase 1, which includes oxidation, reduction, and hydrolysis. That's in comparison to phase 2, which is generally conjugation reactions, meaning adding on some sort of group to the metabolite in question. And both of these phases are controlled by pharmacogenetics. So genetic variation has influences on both phase 1 and phase 2 metabolism. Phase 1 metabolism, which really concentrates on CYP enzymes, which is really the main kind of clinically actionable pharmacogenetics that we see currently. And we'll be talking mainly about CYP pharmacogenetics. In this pie graph, it's broken up by the percentage of drugs that are actually metabolized by a given CYP. So see here, 3A4 is about 30% of drugs, 2D6 is about 20% of drugs, 2C19, 7%, 2C9, 13%, and so on and so forth. And then for each of these CYP enzymes, they're telling you the types of things that influence the enzyme's function. So let's take a look at CYP2D6. Inflammation can influence the function of CYP2D6 by decreasing its efficiency, while polymorphisms, which we will learn about, can either decrease or increase the enzyme's ability to metabolize metabolites. Another consideration, let's look at CYP1A2, which is about 9% of drugs. It can be induced by other drugs. Inflammation also decreases it. Cholestasis decreases it. Age increases the metabolism. There's a question of whether or not sex can actually influence the enzyme's function. And then again, polymorphisms. So as you go through this entire pie chart, you can see the things that potentially influence it and the things that don't, with the bold being the things that are bigger influencers of those enzymes. So one thing I will say is that CYP3A4/5 is about 30% of drugs. So almost 1/3 of all drugs go through 3A4 and 3A5. However, to date, we have not found very good variants within either of these subtypes of CYPs that have clinical effects. So that's a sad face. We're hoping someday we figure that out. Maybe it's specific haplotypes. Maybe it's a combination of CYP3A4 with other genes that we're not sure. But to date, there's really no super strong clinically actionable evidence with CYP3A4/5 genetic variation. Let's take a look at what a theoretical effect of pharmacogenetics is on phase 1 metabolism. So let's say we have a graph here. On the x-axis, we have time, and on the y-axis, we have drug concentration. In this scenario, we can have 3 different metabolizing capacities. So let's say the first one we'll do is what would be considered a normal metabolizer. And that might look like this, starting giving on the drug first day. So as you can see, I'm doing more of what would be a elliptical kind of looking like sound waves going up as the drug concentration increases. And why is that? It's because I'm showing you an oral drug where we give a drug for a day, it goes up, and then it starts to come back down. We give the drug again, starts to go up, goes back down, give the drug again, and so on and so forth until it starts to taper off here because we're reaching steady state. So this first one would be what would be considered an extensive or normal metabolizer. Now let's do a scenario where someone is a poor metabolizer and see what that looks like. And that would look like something quite like this, where we would have, this one would be a poor metabolizer. And then finally, what would happen if we had someone with rapid metabolism? That would look probably something like this. So you have 3 different metabolizing types. You have the extensive normal, you have the poor metabolizer, and then you have the rapid metabolizer. And so why are these all important? Because we have different windows when we give drugs. For example, this middle window where the extensive or normal metabolizer ends up could be considered the therapeutic window. Up here, this person would have decreased metabolism and they're above the therapeutic window, and then one would guess they'd probably have side effects. And this person would have increased metabolism, suggesting that they might not have any therapeutic benefit from this drug. So as you can see here in this simple demonstration, someone who has what would be non-normal or extensive metabolism could have too high of drug levels or too low of drug levels. All this talk about critical genetic variation and its effect on things like metabolism, there's a really important note that I want you guys to know, and that is the following. Even though pharmacogenetic effects on PK can be large, this does not equate to an increased or decreased risk of toxicity or non-benefit. And why is that? Again, if we remember back to our previous slide, it's all about that therapeutic window. So obviously, if you have a really wide therapeutic window, you could have a lot of genetic variation that may not have much of an effect. Versus if you have a really small therapeutic window, your genetic variation can easily land you on the upside or the downside of that, like we saw in the previous chart. And there's even examples like this out there in the literature. For example, paroxetine has pharmacogenetic information in the drug label as approved by the FDA. However, there is no clinical effect. So in the label itself, it says that certain metabolizers of paroxetine can have severely elevated concentrations. I believe it's anything from 5 to 10 times. However, there's no clinical effect yet that's been observed in the literature. So always remember that. Although pharmacogenetics can have very large effects on metabolism, it really depends on the drug specifics and really can depend on that therapeutic window. So let's switch gears from pharmacokinetics to talking about the pharmacogenetic interactions with pharmacodynamics or drug transporters. So transporters can be involved in all different stages of the absorption, distribution, metabolism, and excretion of drugs. Two examples here I just want to highlight are P-glycoprotein transporters are involved in the gut absorption of drugs and in the passage of drugs through the blood-brain barrier. Another common example of PD that we think of is the OP transporters. And these transporters actually increase uptake in the liver for elimination from the body. So you can have pharmacogenetic variation in both. On drug receptors as well. So these variations, these genetically induced variations in drug receptors can cause, for example, increased or decreased on or off target effects. So what this is referring to is you might have more affinity for a receptor, decreased affinity for a receptor, or if you had genetic variation in some sort of homologous or similar receptor, you can actually have your drug molecule bind to a different receptor that wasn't really intended for. Additionally, you could have an increase or decrease amount of receptor available for the drug to work at. Obviously, if you had lower receptors, maybe your drug actually isn't able to produce enough therapeutic benefit because there are enough receptors on board to cause the mechanism of action. And what's a real example of this? It's in a gene called SLC6A4, and it has these long and short versions. And so what is SLC6A4? It's a serotonin receptor. And what they found is that when you have a long or short version, which occurs in the promoter region, this is associated with decreased transcription, decreased protein product, and therefore you don't have as much of the serotonin transporter available for a drug to work at. So you might have decreased drug effects. And there is some literature out there that supports this specific genetic variation with, for example, SSRIs. All nomenclature for pharmacogenetics is defined by the Human Genome Variation Society, so the example we're going to look at is for generic SNP nomenclature for the SNP reference SNP number RS1801133. And that would, if you were to look this up in one of the databases we're going to talk about later, that would look like this. And so SNP nomenclature has very specific parts to it. It always has a gene name. Then it has some number that defines the relative position of the variant. And I say relative because it's relative within the gene. So usually it's some sort of number that's saying it's X amount of base pairs away from a transcription start site or within a certain exon, something like that. So it's always a relative number. And then finally, we're getting to the information that's really of interest, and that's what are the alleles? The first allele, or whatever symbol's put here, is the wild type, while the second is always the variant. So the alligator sign always eats the wild type allele. And so that's an example of a SNP nomenclature. Now sometimes you'll see alternative forms of SNP nomenclature. This isn't as common anymore because people are really adhering to the Human Genome Variation Society's standards, but you might still see it. So let's look at a few examples. So the first example I'll show you is this one. This one is actually pretty common. So what it does is it puts the wild type before the number and the variant after. So the same, it's still consistent though. It's always the wild type allele first, followed by the variant allele. Another way sometimes you see it, it says the following. Anybody guess what this variant is here? It's actually telling us that this is a non-synonymous SNP, where it changes an alanine at position 222 within the polypeptide to a valine. So very less commonly, but sometimes you'll see the genetic SNP actually telling you what the change in the amino acid is. Then finally, you'll sometimes see the shorthand of the amino acid change. And so that's an alanine, which is the wild type form, followed by a valine change. Let's look at a star allele nomenclature, which is the bulk of what we're going to be working with in class. So star alleles are used not only for SNPs, but other genes as well, and they're really used to show composite genotypes. And where did we learn about composite genotypes? In those haplotypes. So it's really showing you some sort of combination of SNPs. Many times they're actual haplotypes that are making up the star alleles. So let's take a look at what a star allele nomenclature looks like. So just like the previous one, it always starts with the gene name. For SNPs, it actually gives the parent and the subfamily within the gene. And then you always get two alleles showing up. Allele 1 and allele 2. So this is where it's different than SNP nomenclature because it's not telling you the variant change. It's telling you an allele that's defined by a certain composite of SNPs. And we'll see that in a few slides. So just remember that star allele nomenclature shows you either one or both of a person's alleles. So for example, in general, the wild type allele for all SNPs and star nomenclatures is star 1, star 1. And then from there on, it's a little bit different depending on the SNP, but most of them, as you go up in numbers such as 2 or 3, you're getting worse functioning of the enzyme or protein in question. So why is nomenclature so important? It's because it tells us all about that phenotype. So let's take a look back at our MTHFR example here. So MTHFR is involved in folate metabolism. So the folate cycle kind of goes in a circle. And what happens is you have folate that gets activated by MTHFR into an active folate. Then folate interacts with other cycles like the homocysteine cycle and other things like that. So let's see what the phenotypic effects of this genotype that we already just looked at a few slides ago. Let's see what the actual phenotypic effect is. So CC is the wild type. These people have normal folate metabolism and they have normal homocysteine levels. CT would be the heterozygous genotype. It carries one dysfunctional allele. So it has decreased efficiency, this MTHFR enzyme, of 60%, so pretty big. So it has slightly increased homocysteine. It actually has decreased B12 and decreased folate. So if we have 60% reduced efficiency with the heterozygote, what about the homozygous recessive that has two dysfunctional alleles? And what you'll see here is an 80 to 90% decreased efficiency, which leads to fairly large increases in homocysteine levels and fairly drastically reduced B12 and folate levels. So we can see here the genotype can end up telling us a lot about someone's phenotype in an example like this. Similarly, the star allele system will also tell us a lot about a given enzyme's or protein's phenotype. So we're going to highlight the SIP system again because that's what we're working on so much in this class. So a poor metabolizer is defined as having 2 dysfunctional or non-functioning, at least 1 allele. So very often, this is defined as numbers greater than or equal to star 2. However, there are exceptions to this rule, specifically with SIP 2d6 that we'll see. But the overall point is that a poor metabolizer has 2 dysfunctional or at least 1 non-functioning allele. And where do these non-functional or dysfunctional alleles come from? That goes back to the beginning of the lecture where we learned about non-synonymous SNPs, about stop SNPs that code for a stop protein, copy number variations that decrease the function of protein, something like that. So naturally, you probably could guess an intermediate metabolizer has generally at least 1 decreased functioning allele, but no loss of function allele. So many times, this one will be some sort of combination of the wild type, star 1, with one of those reduced functioning alleles, which it generally starts, again, at star 2 or bigger, with the exceptions that I just talked about. Extensive metabolizer is also called normal metabolizer, and this one's always defined by star 1. And this is the same across any sort of star allele system, even something other than CYP enzymes. And then rapid metabolism is not present in all CYPs. It's actually only present in 2 different CYPs. 2C19 and 2D6. So for 2C19, you have to have at least 1 star 17 allele, versus 2D6, you have to have some sort of duplication of star 1 through star 3. And we'll see those in action later. But in the end, remember that a poor metabolizer for a star allele system has 2 dysfunctional or a fully non-functional allele. An intermediate metabolizer is what the name says it is. It's an intermediate, so it's a combination of an extensive metabolizer and a poor metabolizer. An extensive metabolizer is always star 1, and ultra rapid doesn't occur with all star alleles, but the 2 we talk about in class is 2C19 and 2D6, where 2C19 specifically has a star 17, and 2D6 has a duplication, which increases the amount of product that's created for 2D6 of star 1 to star 3. And I should mention that this specifically looks like this, where it's star 1 times a number, or star 2 times a number. And the times number is telling you it's a duplication. It all comes back to those composites, meaning multiple combination of SNPs, and possibly haplotypes. So let's see some examples in here. So you can see here the star 1A that has no nucleotide changes because that's the star 1. You can have actual subtypes. CYP2D6 is a highly polymorphic gene, which we'll learn about. So you can actually have subtypes within star alleles, but let's just pick some out for highlighting sake here. So let's look at, let's say star. So in star 2A, it has this 1661 G to C. So at position 1661 in CYP2D6, it changes a G to a C. But you also see that shows up in other subtypes of star 2. It shows up in star 2E, star 2F, and so on and so forth. And if I had enough room to put a full table in here, is what you would see is that particular different star alleles, they have carryover in what SNPs they have, but it's the combination of SNPs that define certain star alleles. And also of interest here, you can see this star 3. It actually is a deletion SNP, so it's at the position 2549. It's an A that gets deleted. That causes a frame shift mutation, meaning that it actually shifts the codons to the left, causing a truncated protein, causing a non-functional allele. So this one would be a poor functioning star allele for CYP26, but we'll get into that more.